At mAbxience, we develop, manufacture and market biosimilars, mainly monoclonal antibodies, using the most advanced technology and cutting-edge analytical methods in our processes, following strict international regulatory guidelines. First, an in-depth analysis of the reference product is carried out to obtain all the possible information about its structure. This is a stage in which a considerable amount of time is invested, guaranteeing the best results. In the transfection phase, once the DNA sequence that corresponds to the antibody is synthesized, it is inserted into the plasmid. The plasmid with the DNA transfects the cell, where the gene corresponding to the antibody and other genes needed for its translation are integrated randomly into the cell genome and thereby producing the desired antibody. In this stage, called random insertion, a set of polyclonal cells is obtained. These cells produce different quantities of antibodies with minor differences. The development of the cell line phase starts. During this phase, the cells are isolated and analyzed using different techniques until only the one that produces the most similar antibody to the innovator in terms of quality remains. This is when it is frozen and the cell bank is obtained. This is the basis for the following phase, upstream process. In this phase, the vial is thawed and cultivated, first in flasks by shaking, before being put into the bioreactors. The technology used is single-use, which provides greater flexibility for production and prevents cross-contamination via the use of disposable materials. In the bioreactors, nutrients and physicochemical parameters are vital for cell growth. As such, temperature, pH or oxygenation are strictly controlled during this phase. In the bioreactors after the production phase, all remaining impurities are eliminated in the purification or downstream process using chromatography columns and filters, which, by means of different resins, enable all the undesired components to be discarded whilst retaining only the antibody which is the most similar to the reference product. Lastly, during the formulation phase and before filling the vials or syringes with the final product, the antibody is added to a solution containing the necessary excipients to stabilize it and make it suitable for patients. This is how mAbxience develops and manufactures biosimilars, with focus on quality throughout the whole process, to achieve that each product delivered to patients is highly similar to the reference product. We call this fingerprint-like biosimilars by design.